Welcome to Let's Get to the Marks. Grab some paper, grab a pen, pause the video, have a go at the question, then unpause and watch the walkthrough. This is on proteins and amino acids. All right, 8.1, describe the biochemical test to confirm the presence of protein in a solution. So what you should be looking at here for your answer is the Biuret's test. And you want to use the Biuret's reagent, which you add in a test tube to whatever sample you're testing. Now, you can alternatively say add copper sulfate and sodium hydroxide. You'll have heard of those before. Copper sulfate is a blue solution which gives Biuret's its blue color. Now, we don't need to warm it, but when you add it to the sample, if the sample contains amino acids or proteins or peptides, it's going to turn from blue to purple. I like to think of P in protein and purple for the colour it turns. You can say pink or lilac or violet, and that would get you 2 marks. 8.2. A dipeptide consists of 2 amino acids joined together by a peptide bond. Dipeptides may differ in the type of amino acids they contain. Describe 2 other ways in which all dipeptides are similar and one way in which they're different. So what I'm doing here is I'm drawing out the structure of a dipeptide. Now it tells us in the question that they're joined together by a peptide bond, so you should be able to work out which bit of this structure is the peptide bond. And you can't talk about that because it's already said it's in the stem of the question. But what 2 other ways are the 2 dipeptides or all dipeptides similar? So what things will all dipeptides have? Obviously, excluding this bit I've circled here, which is the dipeptide bond. So they've all got an NH2 at one end. Do you remember what that's called? And they've all got a COOH at the other end. Do you remember what that's called? So they all have an amine group or an NH2 group, or you can call it an amino group / amine group, AMINE. That's the NH2 group. You can say NH3 plus when it's a zwitterion. They all have a carboxylic acid group at one end, or you can say the carboxylic group. You can write the formula COOH, or you can write COO minus, like a carboxylate group. When it's a zwitterion, it loses a hydrogen. So those are all acceptable to say, and that would get you 2 marks for the 2 similarities. But there's other similarities here as well. What elements do they all contain? What do all elements do every single dipeptide contain? Well, there's 4 atoms in a protein that are consistent amongst all proteins and all amino acids, and it's nitrogen, oxygen, carbon, and hydrogen. So they have those atoms or elements. And all dipeptides will contain 2 R groups, so that's another similarity. Now, what's the difference that all dipeptides will have? The real only difference is in the R groups. You can have different R groups. Of course, you could have the same R groups. You could be a dipeptide made of 2 glycine amino acids. But for example, the R groups can be different because you could have something like cysteine that contains sulfur atoms. Excuse my spelling there, that's not how you spell cysteine. And you could have glycine, which doesn't have a sulfur atom in it. So the structure and the constituents of the R groups could be different. All right, last part to the question. A solution contained a mixture of 3 amino acids. That's key. They've put it in bold there. You must circle these in your exam because it means it's part of the answer. A scientist passed on an electric current through the solution to separate the amino acids. She placed a drop of the mixture on some filter paper, attached an electrode at one end of the paper and switched the current on. She switched off the current after 20 minutes. We can see the spots of the amino acids in new positions. Explain what the positions of the spots in figure 6 show about the amino acids. Pause the video, have a go. Okay, you can see I put a plus and a minus at the start and the end. So we know the spots have moved towards the negative side. So what does that tell you about their charge? Those amino acids must be positive because they've moved off the line. If they had no charge, they'd stay on the line. If they were negative, they'd move towards the positive end. So they've been repelled by the positive electrode and attracted to the negative electrode. So that's going to get you a mark for explaining why they've moved that way. What can we tell by the fact that they haven't moved the same distance? There's 2 amino acids there that are separated. Well, we can tell that 2 of the amino acids must have different masses or different charges as they have moved at different rates through the filter papers, different speeds through the filter paper. So after 20 minutes, they haven't been going at the same speed. One has gone further than the other, so it must be lighter, or one has travelled slower, so it must be heavier. Now, the third mark is the hardest mark. Remember, we circled 3 different amino acids. How come there's only 2 spots? If there's 3 amino acids, why is there only 2 black spots? And that is because it tells us that 2 of the amino acids must have the same mass and the same charge because they have moved the exact same distance. So we can only see 2 spots. It means one of these spots, there are 2 amino acids in it. So obviously that mark is quite hard to work out, but they put the little clue in bold at the top there, so you always got to watch out for that. So what I'm going to do here, I'm going to draw the black spot on the line, and what this shows us is that this spot moved. I'm just trying to select it. This spot moved the same distance as one of the other spots, so it appears like there's only 2 amino acids, but we were told there were 3, which means one of these spots contains 2 different amino acids. That must have the same weight and mass. Okay guys, thanks for watching. Let's get to the marks.
You're still here? It's over.